Hello everyone, this is our first video for biology discussion NEET 2021 exam. So first question 101. So cells with active cell division capacity and tissue having all cells similar in structure and function, tissue having different types of cells, and dead cells with highly thickened walls and narrow lumen. This will be dead cells, highly thickened wall and narrow lumen. It's a option. It's a option 2. That is first one. So dead cells, dead cells is scleroid. Dead cells will be scleroid. So dead cells is scleroid. C will be 1. That is C will be 1. Tissue having different types of cells, that is vascular tissue. Cells with active cell division capacity, it will be meristematic tissue. A2 B4, A2 B4, C1 and D3. So answer will be option 2. Now question number 2. Which of the following is an incorrect statement? Option 1, nuclear pores acts as passages for proteins and RNA molecules in both direction between nucleus and cytoplasm. Yes, this is correct. Nuclear pores acts as a passage for proteins, and in case of eukaryotic organisms, there is a nuclear pore is there. Nuclear pore complex and this through nuclear pore complex, RNA molecules and proteins also RNA molecules and proteins also pass in both direction between nucleus and cytoplasm. Now, microbodies are present in plants and animal cells. Microbodies are present in plants and animal cells. Yes, this is also correct. The perinuclear space forms a barrier between the materials present inside the nucleus and the cytoplasm. Perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. Yes, this is also correct. So, mature sieve tube element possesses a conspicuous nucleus and usual cytoplasmic organelle. No, actually, mature sieve tube elements lack of nucleus. Sieve tube actually lack of nucleus. That's why it's attached with companion cells for the helping though it is a living cells, but it's attached with companion cells. So answer will be option 2. Now 103. When gene targeting involving gene amplification is attempted in an individual tissue to treat disease is known as gene therapy. When gene targeting involves gene amplification is attempted in an individual tissue to treat disease is safety testing, biopiracy, molecular diagnosis. Only one option is there, that is gene therapy. So answer will be option 3. 104. During purification process for recombinant DNA technology, addition of chill ethanol precipitates out, that is DNA. Chill ethanol, when we actually extraction of DNA, when we isolate DNA from any tissue, any cells, even in blood cells, we use ethanol precipitation, and precipitation actually used, and DNA actually precipitated through this method, right? And now 105, protoplast fusion and plant tissue culture, meristem culture, micropropagation, and it will be option 3. That is A 2 promote pomato, B1 that is totipotency, C4 meristematic culture, that is virus free plant, and D micropropagation, D3 somaclone. Answer will be option 3. Now 106. When the centromere is situated in the middle of a two equal arms of chromosome, the chromosome is referred to as, it's called metacentric chromosome. When the centromere is situated in the middle of the two equal arms of the chromosome is referred to as metacentric chromosome. The factors that leads to founder effect in a population is called as genetic drift. Founder effect in here, on genetic drift is the major important factor. Now 108. Which of the following is a correct sequence of steps in PCR or polymerase chain reaction? First is that in PCR step, what we have actually done. First, we have denatured the DNA segment so that DNA strand gets separated. That is called denaturation. Then annealing and extension. So answer will be option 2. 108, option 2. Now 109. In spite of interspecific competition in nature, which mechanism the competing species might have evolved for their survival? In spite of interspecific competition, which of the following actually predation and interspecific competition nature, which mechanism competing species might have evolved for their survival? Only for, have to avoid intercompetition. That is major important part is that is mutualism. So answer will be option 4. A typical angiosperm embryo sac at maturity is it contain in case of in mature it's have a 8 nucleate and 7 cell. In case of 8 nuclei, we know that there is a antipodal cells, synergid cells, and in polar nucleus is 2n. So there is a 8 nuclei is there, 8 nuclei and 7 cells is there, right? One is diploid. That's why 7 cell. So the site, so answer will be 110. Answer option 2. 8 nuclei, 7 cell. The site of perception of light in plants during photoperiosm, it will be leaf. So answer is option A, 1. And 112. Which of the following plants is monoecious? Monoecious plant is Kara. Kara is the monoecious. Kara is monoecious. Option 3. So 113. 113. So Christie thylakoid centromere's sister knee. Christie is the finger-like projection, membranous projection and infolding of the in mitochondria. So A will be 3. A3, only one option is given, that is 113 is option 4. Christie is the infolding in mitochondria. So answer will be option 4. Cohesion, adhesion, surface tension, guttation. 114, it will be option 2. That is cohesion A 2, that is mutual attraction water molecules. When the actually transport of water transport actually takes place, and that time the water molecule attract by each other by bond is called cohesive force. Another is the adhesive force is there, and adhesion. So A will be option 2. A2 114. A2. B adhesion. Adhesion is the attraction towards the polar surface. C is the surface tension. It will be 1, more attraction in liquid phase. And D guttation is the 3. That is water loss in liquid phase is called guttation. Now 115. Lenticell, cork cambium, secondary cortex and cork. 115, it will be option 3. 115, it will be option 3, right? So A3 lenticell, exchange of gases. B cork cambium, it will be 1 phloem, phelogen. C secondary cortex, it will be pheloderm. And D cork, it will be 2 suberin deposition. The term used for transfer of pollen grains from anther of one plant to the stigma of a different plant, which during pollination bring genetically different and types of pollen grain to the stigma, is called xenogamy. So 116, it will be xenogamy. So which of the following not an application of PCR polymerase chain reaction? During polymerase chain reaction, we actually amplify the DNA segment, used for molecular diagnosis, used for gene amplification, used for detection of gene mutation, but not for protein isolate purification of isolated protein. So answer will be option 4. It is not related to protein, it is only related to DNA amplification, gene mutation, right? So answer will be option 4, purification of 117. 4. The production of gametes by parents from formation of zygotes, the F1 and F2 can be understood from a diagram called, it's called Punnett square, which those actually we use the rectangular square is called Punnett square, as the Punnett first discovered it. That's why it's called Punnett square. Which of the following algae produce cara green? carrageen. So 119, it will be 4. Red algae produce carrageen. Amensalism can be represented as amensalism is a minus zero interaction. There is different type of species interactions are there, like commensalism, amensalism, parasitism, predation, competition. There is different interactions are there among the mutualism. Mutualistic relation is a plus plus relation. Commensalism is a plus zero interaction. Amensalism is minus zero interaction, right? So parasitism is a minus plus interaction. So this type of pcc pcb interaction is there. So here answer will be 120. It will be 2. A species A minus and species B 0. Which of the following stage of meiosis involve division of centromere? It will be anaphase 2. Now 122, question number 22, complete the flow chart of the central dogma. We know that DNA to DNA is called replication, DNA to RNA is called transcription, and RNA to protein is called translation. So answer will be option 4. 112 option 4. 22 option 4. 23, mutation in plants can be induced by, it will be gamma rays. So 23 it will be 4. 24, that is we know that in equation gpp equal to npp plus r, gpp equal to npp plus r. So 124 it will be gpp equal to npp plus r. 24 it will be 1, that is respiration loss. R represent respiration loss. 125, plants follow different pathway in response to environment or phases of life to form different kinds of structure. This ability is called plasticity. 125 it will be option 4. The plant hormone used to destroy weeds in a field is a 2,4-D. 2,4-D is the natural, it's a artificial weedicide. Weedicide we actually used for destroy of the weeds in a field. So 127, which of the following statement is not correct? Pyramids of number, actually there is a two inverted pyramids are there. One is pyramid of biomass in C, generally inverted. Pyramid of biomass generally C is generally upright. So answer will be not correct is this option 3. Option 3 is the incorrect. 127 option 3. Gamma are present in 128, it will be liverworts. It's some liverworts are there. 128 option 1. Now 129, the first stable product of CO2 fixation in sorghum. Sorghum is a C4 plant, so it will be oxaloacetic acid or OAA. Which of the following algae contains mannitol as reserve food materials? 130, it will be 2. That is ectocarpus have a mannitol. And DNA stand on a gel strain with ethidium bromide when viewed under UV radiation appear as, it will be bright orange band. Actually, we use ethidium bromide for visualization purpose, and ETBr actually acts as an intercalating agent. So 131, it will be 3. That is bright orange band. Didelphous stamens are found in pea plants. Which of the following are not secondary metabolites in plants? Basically, amino acid and glucose, they are not secondary metabolites, they are primary metabolites. So amino acid and glucose are primary metabolites. 113, it will be 3. So 134, the amount of nutrients such as carbon, nitrogen, calcium present in a soil at any given time is known as standing state. Another is the organic materials. If the amount of organic material present in a particular given time in a soil is called standing crop. So inorganic materials, that is carbon, nitrogen, phosphorus, calcium, these are called standing states. So answer will be option 4. So next is 135. 130, genera like Selaginella salviana produce two kinds of spore. Such plants are known as 135. It will be 1. That is heterosporous. Now section B, plasmid. 136, plasmid DNA PST1. It will be 136. It will be option 2. That is, it will not be able to confer ampicillin resistant to the host cells. Actually, it is from biotechnology question. In some members of which of the following pairs family pollen grains retain their viability for month after release? 137, it will be option A. Option A, JC and Leguminosae. Okay. Poaceae and Leguminosae, Rosaceae and Leguminosae. So 138 answer will be option 3. 139 it will be option